With this, we proceed with the keynote speech of the day. I would like to invite on stage Dr. Sunil Arora to chair the session. He is known for his specialization in immunology of infectious diseases, cancer stem cells, and mesenchymal stem cells for rheumatoid arthritis, T-cell epitope-based vaccines for visceral leishmanias, and dendritic cell dysfunction in HCV genotype 3 non-responders. Dr. Arora received the Indo-US UNAID DBT Young Investigator Award in 1991 and holds memberships in various professional bodies. He has made significant contributions to publications in immunology, infectious diseases, and parasitology. We would also like to invite Dr. Salunke, and continuing with his introduction, he has had a distinguished career as the director of the New Delhi at ICGEB and as the executive director of the Regional Center for Biotechnology. Dr. Salunke's research focuses on unraveling the physiochemical principles of molecular interactions in non-self discrimination, with notable research impacting the understanding of antibody degeneracy and immune recognition. His work involves molecular mimicry, carbohydrate peptide mimicry, and carbohydrate porphyrin mimicry, shedding light on diverse facets of immunology. I would request Sir to deliver his keynote on connecting immunology and pharmacology.
So, good morning, everyone, and thank you very much for inviting me here. Because my job is already done, I don't have anything to say, but I'll take this opportunity to say a few words because I have been invited here before Dr. Salunke starts his discourse. You have already known how distinguished scientist he is, and he's presence of such people actually motivates. So that is the major factor why we have very senior people here. But as everybody on the inaugural function was speaking about importance of immunology being connected to the other allied subjects, I would like to say a couple of things. Is that immunology was used to be a simple subject somewhere, microbiologist was doing some serology, we will say that is a immunology laboratory. But now the immunology is not that. It has come in a big way from diagnostic to mechanism of disease for treatment and now therapeutics. And everybody spoke about CAR T cells and T cell specific monoclonal antibodies, they all have come. So connecting immunology with the audience, Indian Immunology Society has conducted so many, such kind of courses. We have been doing that, and this is the first time that at Nirma, Dr. Sarath has started this kind of program for the pharmacy students. And it is very important that all the pharmacy students should understand what is immunology. And with that word, I would invite Professor Salunke to please start his.
I would like to thank Dr. Dalai for giving me privilege to speak to you first. So nobody else would have spoken what I am going to speak before. So I will have that advantage, but other speakers may hopefully elaborate on what I say and not necessarily I will be taking their words in its entirety. I think a lot was being discussed about connecting immunology and pharmacology, and I'm not really expert in both the areas, but I indulge into immunology because about 35 years ago I joined National Institute of Immunology, and as they say, while in Rome, be like Romans, so I started learning immunology. Practically, I learned my immunology from my students, and since Dr. Dalai, while inviting me, said, I need to give a keynote address and it should connect immunology with pharmacology, and I have made my best attempt to do so. So immunology and pharmacology actually seem to be entirely unrelated subjects. One is, I think everybody has talked about this. Immunology is essentially a study of immune system, its components, and its applications. And pharmacology is focused on interaction between drugs and human body. And these two would apparently look very independent. So these two areas do have a very strong overlap. And on one hand, immune system gets affected when you take any drug. And that is very obvious, and to study that itself is very important, as Sanjeev Kumar. So immune response to pharmacological intervention is one aspect, which is a consequence of actually pharmaceutical interventions in the human body. And that is very obvious, and that is one of the major issues that one has to study when one actually tries to make biologics based on antibodies. And the other is how to exploit the immune system in terms of making pharmacological applications. And that also, again, Sanjeev Kumar mentioned that today most of the biologics are antibody based. And so these two are the main connections between the immunology and pharmacology. I'm sure there are many more connections, but I will limit myself to talking about this. And if you're talking in terms of what actually happens when you take drugs in terms of effect on the immune system, one most important is immunomodulation. And in fact, there are many drugs which are designed to be immunomodulatory. They become effective by affecting the immune system. But also, there are immunological consequences which are like hypersensitivity. You have completely excessive effect, and it can be different for different people. There can be immunosuppression, and due to some drugs. In fact, there are drugs that are designed for immune suppression for certain purposes, particularly organ transplant and so on. And there are also situations when you take some drugs, you have cytokine release syndrome, where are large number of cytokine cascades actually come up, resulting in inflammation and so on and so forth. You do have some side effects of drugs that affect immune system in a very indirect way. You don't necessarily know the pathways in which it is getting affected, and that is in some sense like side effects. So all these areas are subjects on their own, and many studies have been done. And I'm sure over the next 2 days, many of these areas will be covered. And I'm not going to talk about them beyond this highlighting. But the other aspect, which is pharmacological exploitation of the immune system, can be looked at in terms of antibody therapy, which was mentioned again and again in inaugural session. And second thing, which is equally important, is vaccine development. And as I said a little while ago, that vaccines has been one of the most important gift of immunology for humankind, and that is probably one of the oldest of medical applications based on immunology that we know, almost 300 years old. So I will give a little bit of overview of these two aspects, because in some way or the other I was connected with these two areas. And central to both of them is essentially antibodies. And in one case, you actually tailor make the antibodies for a specific purpose. And other case, you work with the antigen to make antibodies so that they can be neutralizing the disease that you have targeted for. And considering that, I actually thought what we really need to know, as was mentioned in the inaugural session, that you really need to know physics and chemistry of the antibody molecules. And that also is studied for almost 100 years now. In fact, antibody response, one of the most important property that is highlighted and is important is actually its specificity. And even though antibodies were not identified as a specific entity or not handled at individual molecule level, in 1930, Landsteiner actually showed very elegantly by a very simple chemical experiment that immune response can be extremely specific. As you can see here, what I'm showing you is an aminobenzene molecule, and this experiment he did practically about 95 years ago. And if you have antisera raised against aminobenzene, that will be very specific to aminobenzene. A simple chemical modification of one carboxylic acid group will actually not cross-react. And if you take any of these four molecules, where even there is an amino group on a benzene ring and carboxylate group on three different positions on the benzene ring, chemical formula for all of them is same, but the antisera will not cross-react with this. And this is a very simple assay demonstrated how specific these antibodies can be. And at that time, what was the composition of that antisera was not really known, but it was subsequently identified. It is the antibodies that play a role and as a consequence of immunization. And this mice experiment could not have been taken further in terms of understanding the molecules that are responsible for providing this specificity. And somebody like me who has training in structural biology will not be satisfied with the chemistry as understood in terms of what is biochemical experiment, but want to look at the molecules by themselves. And there is no way one can go any further to understand molecules or to be able to visualize molecules by themselves. And so one really needed to see antibody molecules. And to be able to see antibody molecules, one has to see them as individual entities. And that is where the discovery of monoclonal antibodies was essential. And it is until 1970, one really didn't know how to look at molecules individually. And it is 1970 that group reported single plasma cell clones to be able to be separated, and homogeneous antibody population could be formed. And it was in 1975, Kohler and Milstein actually were able to come up with hybridoma technology, could make these monoclonal antibodies in large quantities, for which they got Nobel Prize in 1984. And so one could do a lot of experiments, biochemical, biophysical experiments using antibodies since 1970, but more so from 1975. And it was immediately within next 10 years that the first monoclonal antibody, which was for anti-rejection purposes, was approved for clinical use by the US FDA. And that is the history which goes further in terms of structural biology. And it was in 1974, the first report of a three-dimensional structure of an antibody molecule that was actually came up. And that provided a very, very nice topology of antibody molecule. Now, this is not based on the report of Porter and Edelman in 1974, but it is evolved over a period of time in terms of details and resolution. And basic topology is actually very simple. What you see as a Y-shaped molecule is essentially made up of 12 domains, which are repetitive units of what is called as an immunoglobulin fold. And this fold is same in all of these. 2 of those in light chain and 4 of them in heavy chain. There are 2 heavy chains and 2 light chains together. This is actually a very complex structure. Even if you want to make a recombinant immunoglobulin molecule, it's not actually very easy to make it because it has an architectural distinction in terms of 4 chains, large number of disulfide bonds, and so on. So the chemistry of the molecule is quite complex. And that architecture has also been demonstrated in terms of this part of the molecule is really the part which is critical for binding to the antigen for antigen neutralization. And this, what is called as FC region, is the effector function molecule of the molecule. And there is a lot of glycosylation in the FC region of the molecule. So total molecule at its exists is about 150 kilodalton, and the repeat unit, the immunoglobulin fold, is about 25 kilodalton. The it was another 10 years. I think by the time we actually were able to see functional state of the antibody molecule in terms of its binding to its target antigen. The antigen here is a lysozyme molecule, and I am showing only the variable region, rather 2 immunoglobulin folds on the tip of the molecule which are involved in the interaction with the antigen. And that shape there, which is of the epitope or antigen combining site of the antigen and the antigen binding site of the antibody, is as evident here from the 3 pictures, where this picture has been pulled out to show the complementarity between the 2. So in some sense, it actually defines the nature of interaction between antigen and antibody. And if I have to show it in a cartoon terms, it is a lock and key. So you can actually, ability to discriminate between self and non-self, which is a physiological property of the antibodies, can be defined in these 3-dimensional structures to be simply a physico-chemical property of interaction between the 2 molecules. It's a chemistry of non-covalent bonding that you can see there. So it's very interesting. Physics and chemistry of the molecular interaction is utilized towards being able to recognize the antigen. A molecule is made which is complementary to a surface on the antigen, which is called epitope, and to that a complementary molecule is made. Whole machinery is genetic, but ultimately it is physical interactions that give rise to this. And you make large number of those molecules on the surface. So if you take this as an antigen, it could be protein or a hapten or even a whole organism like a virus or a bacterium. But you have these molecules go and bind at the individual epitopes, which is highlighted by one of those is highlighted here in terms of what the complementarity between the antigen and antibody is. And as I said, this is self, non-self discrimination as you see in terms of physical features. And that is what seems to be everything that you need to know if you want to understand immune recognition or how antigen is recognized by the antibodies. But it is not entire truth going by this. Although what I'm going to tell you next, this much is sufficient. Just for the record, I want to tell you that the antigen molecule is not as simple as like a rock-like entity as I have demonstrated here. It has properties. Like organisms change, so do protein molecules. They have a flexibility in them. So if antibodies are made against a molecule and a molecule changes its shape, there will be a new set of molecules or these antibodies change their shape also. This is an important puzzle. I'm not going to go into those details. What I'm going to restrict to you is towards saying that the property that the antibody molecules are very complementary to the surface against which they have been made is an important property in terms of pharmacology that you want to know, because what drugs do is actually go and bind to their receptor very specifically and inhibit them. So that analogy is what actually becomes a very important property as far as my talk is concerned. So host pathogen interaction is understandable by this, but it is not the whole truth. It is a lot more complex than this, and I'm not going to indulge onto that except for take this as an example. So considering that antibodies have this property of specifically going and binding to a target region of the receptor, in this case receptor is antigen, but you could actually take it to rather than taking a receptor which you want to neutralize on the bacterium or on the virus, or by using a drug molecule. In fact, rational drug design is a big industry in terms of designing small molecules. It is lot easily done using the antibody approach in terms of this, and towards that, one really needs to understand architectural features. That's why I actually showed you the IgG molecular three-dimensional structure. And looking at that structure, you can see many, many different variations of that structure have actually come up. There are some variations which are designed to increase solubility, target specificity, immunogenicity, and there are others which are actually to miniaturize this molecule. Since the recognition specificity comes only from that tip part, one could actually simply take the one arm of that Y-shaped molecule as a fragment, or even that would be actually 4 immunoglobulin folds. One could take only 2 immunoglobulin folds, which is called as single chain FV region, and link them so it becomes a one continuous chain, easy to make a recombinant molecule, and it will have all the properties of that binding site to the epitope. So the antigen combining site would be all there. But people have tried to make just one immunoglobulin fold out of this to make antibodies. And in fact, camel antibodies are actually single chain antibodies. And one could think in terms of making therapeutic antibodies even using this. So these 3 variations, in addition to that, you have large number of variations in terms of all these properties, variety of reasons. People have studied antibody interaction with the, in terms of human various tissues, and realize what all modification one can use. and those are all combinations and permutations people have tried, and the antibodies that are actually in the market are many of these combinations. And I have shown here the antibody market, how it has grown since first time monoclonal antibodies were actually developed till 2019, and it has gone up almost exponentially. And what are in the red here are those top 10 antibody molecules that are selling. And it is estimated to be 300 billion US dollars by 2025. So it's an important area where you can achieve commercial interest, as in people in Gujarat do, but at the same time do it by understanding fantastic science. This is being what the therapeutic antibody market has grown into, and there are multiple different ways that people produce monoclonal antibodies. I mentioned about the hybridoma technology. It actually is very straightforward. From those plasma cell, one can actually hybridize with cancer cells, and they produce continuously in mouse model monoclonal antibodies. One can actually later make them humanized to look like a self molecule. Subsequently, a big technology that was developed, for which Nobel Prize was given about 5 years back, fast display technology. One could actually put antibody molecules either in variety of size, but most of the fast displayed antibody molecules come in single chain antibody. That is variable region, 2 domains of immunoglobulin fold. And to start with, the immunoglobulin fold that is put there is of size of a human monoclonal antibody. And so you go through screening process to actually identify the large pool of antibodies that are displayed on the phage, and you can actually culture them and screen against the target, and you can get human antibodies. You could actually make human monoclonal antibodies by making a transgenic mouse do the same thing as what you are doing in hybridoma technology. And there is a more new technology called single B cell. The difference between single B cell based monoclonals that you make there is that this is a library that is available of the 10 raised to power 8 or 9 different antibody molecules will exist in this library. Here you take antibody B cell from the patient. So you will have B cells which are exposed to that particular infection that patient has encountered, and you will be able to make antibodies only specific to that particular infectious agent. Whereas in case of fast display, you have that library, you can subject it to any infectious agent and make antibodies accordingly. And whatever antibodies are available in the market actually are available, which are generated from, I think, practically all of these applications. And I'm going to tell you a little more about the fast display technology. So there are, as I said, single chain variable region library, and human libraries are available. People make their own libraries. I'm sure most people these days make libraries for variety of purposes, and advantage with single chain antibody is basically, since it has minimum paraphernalia, but all that is required for binding antigen, it is the ideal kind of thing. And we have been using 3 libraries, Tomlinson I, Tomlinson J, and this is a creative library, which are 10 raised to power 8 to 10 raised to power 9 different antibodies actually put in each one of them. So these libraries, the way they are used are schematically shown here briefly. I will not go into the detail. This is the fast display library. So these are the antibody molecules. You take the antigen or a epitope. Epitope can be smallest part of the antigen, and then screen these and remove the non-specific ones, and those which are specific, in one round you may not get very highly specific antibodies. So you go into 3, 4, in about 4 rounds, you can actually get highly specific antibodies, and then you sequence those genes, that part of the gene which codes for the antibody, and then you can take that sequence, clone, express, and you can purify it. All of this can actually be done in a matter of month if you have practiced the technology well. My students have used this technology in the lab in variety of purposes, but I just want to illustrate you this in a very simple way in case of 2 situations which are very diverse situations. One is in case of making monoclonal antibodies which could have therapeutic applications in case of influenza virus and in case of snake venom. Influenza virus, as you know, is caused by influenza, causes influenza, and there are 2 variants, influenza A and B. This is a negative sense single-stranded RNA virus, and what most of the antibodies that are available generally in the patient will be hemagglutinin antibodies, and the molecule which varies maximum on the surface is the hemagglutinin. So hemagglutinin mutations is what makes influenza virus escape. So you can't make drug, you can't make vaccine. Even if you make a vaccine, after one epidemic, next year you actually need to have another vaccine, and so on and so forth. There are attempts made by people to actually generate antibodies against M2 protein, which has a substantial part of it which is invariant. It does not change, it doesn't have mutation. Some part of it changes. But so what my student did is take that exposed region of this M2 protein, which is an ion channel. It actually, it's a proton channel, and he picked up this region which is exposed, cytosolic part. I mean, it's not buried part, but exposed part, but it has minimum variations, which is about 8 amino acid stretch, and then screen it against the FAS library, and found, after doing all this exercise that was there in the previous slide, he was able to make about 4 antibody single chain FV domain, and 4 of them he could found, and he could see they bind with the affinity of 10 raised to power -7. In 2 cases it is sub-micromolar, and this is micromolar range. So there are reasonable antibodies to begin with, at least to prove whether it works or it doesn't work. Then we took help from Dr. Maitreyi Rajala in JNU, who also works extensively in influenza virus infections, and carried out two, three assays, but I'll actually describe you one assay in a very simple term. This is influenza virus propagation. So these are the cells, these are cell lines which were used for this purpose. And if you propagate them in an appropriate medium, this is the virus, and the virus enters in it and actually replicated, and it comes out in the supernatant. So virus will be there inside the virus will be there inside the cell as well as outside the cell. If you have an antibody that is working on the virus, when you add there, it is possible that the virus may not be there in the supernatant, and virus is only there in the virus if virus is doing something to that ion channel. And it turns out of the 4 antibodies that were made, one of them actually showed no virus in the supernatant, but there was virus as much as what is expected in case of intracellular. So this antibody AU1, which is also among the highest affinity antibodies, which seems to show actually ability to inhibit replication of this virus. What my student did, more than, this is where being a research scientist makes a lot of difference. We're not so much worried about finding a solution to the problem, but actually to ask question as to how it works and why it works. And towards that end, what he did is he took this epitope, which is an invariant molecule, and tried to make a structure of antibody-antigen complex by X-ray diffraction. And he succeeded in doing that. He took that 8 amino acid peptide and made a complex with the SCFE molecule, and he got a structure which is actually quite interesting. This is a 1:1 complex. There is one peptide molecule bound to one monomer of this, and another peptide molecule bound to another, and it existed as 2:2 complex. And where the two monomers interact but sandwich the pair of the peptides, and that geometry of the two peptides is almost like a coiled coil beta sheet. There are hydrogen bonding here, and actually I'm showing you for the purpose of showing that it is real, what is seen actually in terms of electron density distribution there, which is not very bright, but there is an electron density there. So what it shows is there is an intertwining of these two molecules. And if you go and look at what we know of how does the M2 molecule function, it is very clear. I'm showing here a monomer, but it actually functions as a tetramer for it to be active proton transporter. And when virus assemble, it actually remain active, and lot of understanding of mechanism is available. Whether this dimeric state exists as an intermediate or not, we don't know. But I've highlighted here because our peptide exists as a dimer, and effectively the region of the peptide we had taken for this purpose is this region. And so two antibodies, single chain antibody molecules bind to the peptide and dimerizes. And once these two molecules dimerizes, this in physiological situation, when virus assembly is taking place, there is no way these SCFE molecules will make tetramer formation take place. So it is the inhibition of tetramerization that leads to stopping the virus assembly. And that's why you actually don't see virus in the supernatant. that is what actually is written there. So this antibody against the invariant part of the molecule, which is exposed out, is interfering in the assembly of the virus and thereby restricting the viral replication. Other example, as I said to you, is of making antibodies against snake venom, and we had taken cobra, snake venom, and viper. We tried to get these venoms from sigma, but we are restricted in terms of importing snake venom. So my student went to a snake farm of Chennai. He couldn't carry it by air, so he had to go by train and bring it. It was a very interesting adventure for him. After taking all the wildlife departments, approvals, and so on and so forth. And instead of going in a reductionist approach where we understood molecular structure of M2 and designed a region which we want to target our antibodies, in this case, we actually approached the problem exactly opposite. We took the whole snake venom, which has some 200 molecules, and actually screened those 200 molecules in case of both the venoms to the FAS library. And we were able to get quite a few antibodies. I'm actually showing 7 antibodies here, and out of those, then we took 7 means 5 against naza, which is cobra, and 2 against viper, which is Echis carinatus. So hereafter I am naming N with the number based on the screening experiments, N for naza antibodies and E for Echis carinatus. Echis carinatus is viper. And this ELISA, so we actually discarded one of these right away, which has a lowest binding. So N216 we didn't take further, but what we did is we now took these 6 antibodies and did screening of the whole venom to try and see which component of the venom they bind to. And so for that we had a kit to do pull down of the complex between these 2 molecules, which basically is a specific cobalt binding to his tag, and all these antibodies are with his tag. So I will not go into those details to show this as an intermediate step, and then subjected these to this pull down to a mass spec. You don't need to try to read this, but the summary of this is essentially we were able to identify which toxins each of these 6 antibodies bind to. So 2 of those, N199 and N219, bind to metalloproteinase and nerve growth factor. There are also components of this, but the major component is phospholipase A2. And 2 of the naza and 2 of this Echis carinatus were bound to the PLA2. And then we took these antibodies for mice experiment to check survival of mice, whether these antibodies will prevent or reduce. And what it turns out, in case of naza 2 antibodies, one of them, 194, actually may improve the life for animals by about 50% of them can survive for about 1500 hours. In case of N248, actually only 16% of there was no synergistic effect in this case. I'm not showing data for the Echis carinatus, but only thing I'll tell you, both the antibodies work, but not as good as 194. but when you add them together, there is a synergistic effect that arises from them. In this case, there was no synergy, but this 194 worked very well. So starting from the whole venom, by screening the fast display library of antibodies, we were able to identify antibody, at least one, which can improve the life expectancy for mice by 50%. In other case, when we take two antibodies, we could use those together to improve it to be about 40%. And my student went further, his name is Amit Kumar, and did some of the pathological experiments, and he probed myotoxicity and hemolysis. In case of the NASA antibodies, they showed reduction in myotoxicity, but there is no effect on hemolysis. In case of Ichthys carinatus, actually one of them showed even reduction in hemolysis. So there is a effect biochemically manifested in terms of why there is an improved life for mice here. So we were able to use the fast display library of making monoclonal single chain antibodies and actually arrive at the antibodies which are able to, we are able to demonstrate in the lab that they function, and we were able to probe in both cases what is the mechanism with which they function. I will actually stop about the antibodies here and go to talk little bit about the vaccines. And as you know, vaccine is effectively a mimic of an infectious agent. It has all the properties that can be recognized by the immune system, but it should not have infectivity associated with the immune system. Then that would be a perfect vaccine. And so they are essentially pharmacological interventions designed to stimulate the immune system without giving the infection. And that actually is a fantastic idea which was discovered many, many years ago. And you are basically making immune system of the body more energetic. There are a lot of vaccine designs that have actually been used, and all of them seem to work. And in fact, many of them are actually used in case of SARS-CoV-2. The very first one was the one which essentially to inactivate, say for example, if it is a virus, you just cross-link it or inactivate by heating it. It has an advantage, actually it is a very mature technology, but the problem is it can also, when you do chemical or thermal inactivation, you actually lose lot of epitopes which are relevant in terms of generating immune response. Then you have, within the first generation, you have live attenuated vaccine where your virus works, but it actually has lost infectivity. Then you have subunit vaccine, you just take surface unit which has all the immune epitopes and use that as a vaccine. This became a second generation. It actually was not as good as these in terms of it being able to give an effective immune response, but it actually was very focused immune response. So people actually came up with what is called as virus-like particles, which generated virus-like situation almost like this, but these are intact epitopes on the surface. And then you have vector, which essentially you take a template, or RNA vaccine and DNA vaccine. And as I mentioned, all of these technologies are used. There are advantages and disadvantages in each of them. I probably may not spend time on that. As I said, this is a mature technology and very simple to prepare. This actually makes it intact, but it has a little bit of a risk in terms of not necessarily it is completely attenuated. And subunit vaccine will be very safe, but it actually provides weak immunity, and so on and so forth. Since Dr. Arora has come here, I really have to rush. I will just finish it with this slide. Historically, you can see from 1796, smallpox vaccine, Edward Jenner discovered. And Indian population has been fantastic in terms of accepting vaccination. In fact, over last 75 years, we have done very well in increasing our life expectancy, mainly because of vaccination program. And I travel quite a lot to Europe. I've also given a talk on television to the Italian audiences. And actually, I have discovered the Italians have very, very strong rejection to take a vaccine. And it was evident even during the COVID pandemic. But Indians somehow have accepted vaccination is required for children right from the beginning. And this history has actually helped us save our lives. I have a little bit of anecdote to tell about human papilloma vaccine. That I will tell you and stop there. When human papilloma vaccine was discovered in 2006, I was a scientist at National Institute of Immunology, and they actually planned to introduce it in India. And if you have to introduce a new vaccine in the country, you have to have actually limited clinical trials of that vaccine. To start those clinical trials, there was a meeting of committee to monitor it. Somehow I happened to be invited for that committee meeting, and the group that was going to conduct the clinical trials actually made a presentation on the development of this vaccine. And when they made presentations, first 4 or 5 slides I saw, and I said, wait a minute, these slides are actually taken from data in my paper that I published in 1986. And that paper was this. In Cell 1986, I had actually published a paper on assembly of polyoma virus, not papilloma. And polyoma virus is actually a innocuous relative of papilloma virus, whereas papilloma infects humans causing cervical cancer. Polyoma only spreads in rats. And we had actually understood assembly of polyoma virus. The capsid protein was recombinantly made and expressed, and this is the electron micrograph which actually occurred on the cell cover. And there is one particle which is almost like a virus, and its symmetry and topology was exactly like structure. This graphic picture I prepared based on low resolution X-ray diffraction structure, which practically resembles a particle here. We have done a lot of experiments of assembly and disassembly, and this equivalent of VP1 here is L1 in case of polyoma. So they have used this methodology of making self-assembly of polyoma virus to make similar particles of papilloma virus, and made a vaccine which actually was effective for cervical cancer. And that's when I realized what a loss not to know immunology for 25 years earlier. If we knew this virus-like particle could be used for vaccine development, we would have actually done this for polyoma at the time we did it, papilloma at the time we did it for polyoma. So actually make sure that you learn immunology. So to summarize, what I've actually covered, intricate and dynamic interplay exists between immunology and pharmacology. Well, immunology responds to the pharmacological interventions. Pharmacology exploits the immune system, as is evident from the examples I gave you. Vaccines have been successfully used for a long time, in fact, almost 3 centuries. And monoclonal antibodies are, as you know, being used over a long period of time now. So there is a fantastic connection between pharmacology and immunology. And thank you for your attention.
Thank you very much, Dr. Salunke. Basically what he has covered and he has delivered is that it is basically the discovery of immunological tools which has increased the life expectancy, as also mentioned by Dr. Anup Singh. You know, our grandfather or maybe great-grandfather, they had a life expectancy of only 30 to 40. Sir, can you join me here? So the life expectancy increased with the vaccines. You know, vaccines have really saved so many lives. Until today, it is doing that. You have seen it in COVID, how beautifully the COVID endemic has been suppressed because of the discovery of vaccines. And the second thing which he has mentioned, very important, is the discovery of monoclonal antibodies. So the antibodies which have been discovered, the whole, you know, mystery of immune cells have been delineated with the discovery of monoclonal antibodies. All CDs, you know, we talk about CD4 cells, we talk about CD8 cells. All CD markers are defined because of we have now monoclonal antibodies against them. And then he mentioned the SCFV, which is again a great discovery because that has led to formation of CAR T cells. You know, in the CAR T cells, what we do, we only use SCFV, which is a fragment which can bind to a specific antigen, and that is a antibody kind of structure, but by recombinant DNA technology or recombinant technology, we can engineer that particular molecule into a CD8 cell, which is a killer cell. And now this cell will go and find the tumor and kill that tumor. They are CAR T cells, right? So chimeric antigen receptor T cell. So these are few things which you will know when we will go through all this, because once we go through the immunology and we'll tell you in very basic you know, language what all immune response, how it is generated, how different cells are doing their function, what is the role of antibody, how the antigens are presented, how the immune response is generated, what are different arms of the immune response. So you will understand what is the application part, and these all what he has shown us is a glimpse of all that, what immunology can do to the human life. Thank you very much, sir, for giving us such a beautiful. And I think we should ask a few questions to Dr. Salunke. Any bright question? Yes, please.
So thank you for the very informative and interesting talk. I just want to ask you, like, monoclonal antibody has a big size. That is one of the limitations of for the delivery of the monoclonal antibody in the systems. But as you told about the SCFV, so because I have gone through some papers, so SCFV are having a very smaller size. So does that make it so there is a limitation for it, for the shorter shelf life in the targeted cells?
if you compare with IgG, SCFP will definitely have a shorter life.
Yes, sir.
That is true. But in comparison to what it takes to make IgG, SCFP you can make in bulk much
Yes.
more. So you get that half-life being able to make in large scale, larger quantities. But it is not necessary that you have to restrict to SCFV. One can, once you have identified SCFV and it works in the lab, one can actually make IgG from that by combining with the C, and people have done that.
So I have read some papers in which they have combined SCFV with FC fragment also, and some interleukin molecules also, to increase their size that lies between the whole antibody and the SCFV, so that it can be retained for much longer period of time.
And there are many antibodies which have been made by identifying SCFV, then combining it with FC region, and they have shown that it has improved half-life and it works more effectively. So there are, I think, Sanjeev, there are SCFVs which are in the market by themselves also.
Yes.
But there are people have identify SCFVs and then combined with, enlarged it taking FC component. Some places that effector function of FC seems to come into play also in increasing their function.
Thank you, sir. Thank you so much.
Sir, wonderful talk. I have a few questions, one generalized and specific. You talked about the snake venoms. Generally, when the snake bites happens and toxicity, the time scale, the fastness, compared to, and it does many other mechanism of hemolysis, and as you mentioned. So in that short time scale, when the toxicity happens, compared to the vaccine or any sorts of antibody production inside the cell, and when you making something and exogenously adding, I mean, how that you
That
settle
actually
that?
is a good question, and that remains to be addressed. The important thing is what is available most of the time is this horse antisera as a drug, which is substantially non-specific, so it has lot of toxicity. So people think that if you make some specific monoclonals and combine with it, you actually have an effect amplified, and so it will be much better. So as a combination therapy. And beyond that, if somebody, some farmer is in the remote place, he actually gets a bite, you can't make that available to him in terms of
The time scale,
time. So that question remains, but only if one thinks of vaccination as a strategy, that can be asked. But availability of antibody therapy for remote people is still a big question.
Also, add to that, I mean, it's more than immunology. There are a lot of other biological phenomenon is happening. Second question is for the influenza vaccine you mentioned, say it has a limited time, like 1 year later you have to...
Immune escape.
Correct. So my point is some of the vaccination has a lifelong, validity of, because I'm not sure, I'm a poor immunological knowledge.
Are aiming to get antibodies for regions which are less variability.
So the memory retains for them.
So the mutations don't take, like this part that we are talking about, even if there are mutation hotspots on the virus, this region that we are talking about is not mutation hotspot. There are 66,000 sequences we have looked at, and it doesn't change during that many changes that take place. The most prominent epitopes on influenza virus actually change. There is another interesting thing that I will tell you. This is something which I did not describe because it was not in the context. There is one property of the immune system, that antibodies are generated are actually of two types. There are antibodies which are highly specific, and there are antibodies which have broad specificity. There is a combination of those. So number of mutations that you see, that many are not hotspots. There are very few mutations are relevant. So one can study that aspect and try to design an epitope which even after changes, it is not affecting the antibodies that are generated against it. So the immune escape is a major factor, but in any case, one thing I can tell you, that the most recent vaccines against influenza, even if you have to take annually,
Yeah.
they seem to actually provide you immunity. Once in a year is no big problem for vaccination.
Okay.
Yeah, that thing is true for many viruses, and they keep changing. So I think we may move. You can have discussion later on also. So we can have one more question there. There are so many discussions. You can keep doing it. That's the beauty of the CME program.
Good afternoon, Dr. Salunke. It was privilege to hear you. First question that I have is we talked about NASA 194. So it has established myotoxicity, as we said. But when there is infection of virus, what about the hemolysis that is taking place? Do we have any solution to nullify the hemolysis taking place?
See, as in particularly in case of snake venom, the single antibody will never work as a therapy for multiple reasons. One is there are many effects. Will it influence all the pathologies? In this case, it doesn't. In another antibody case, it actually affects two. Like there is 113, E113, it can reduce myotoxicity, it can reduce hemolysis. We have not tested for neurotoxicity. There is another important question that somebody should have actually asked is this is not the only toxin. Even if you neutralize one toxin, there are several other toxins in snake venom, and they are lethal. So you have to have a combination therapy to be able to be successful in that case.
Thank you, sir. My last question was how did you humanize the mouse antibodies to human by
No,
CD aircraft?
what we made were human antibodies from fast display. Our library was human library. But you are right, I think you have a good point. But we did experiments in mice. So mice antibodies will be rejected by humans. Human antibodies should be rejected by mice. And I suspect this is what we found when we were doing dose dependent experiment. So up to certain dose, it was improving the effect, and after that dose, if you increase further, actually the vaccine itself was becoming detrimental to the mice, and that may be because it is foreign. So we chose a dose which will not be causing death because of the dose. So human antibodies using for mice as an experimental model is problematic.
So humanized antibodies, you asked how you make it. Basically, the antibody has a portion which is constant region and the variable region. In the variable region, you have the complementary determining region, CDRs, which go and bind to the antigen, right? So what people do is they recombinant technology use that they have the human constant region from the human antibodies, IgG, and only the CDR is from the monoclonal antibody, which is raised in mouse. So it causes very less, side effects, as he's saying that the humans will reject mouse antibodies and mouse will reject human antibodies. But if you have the major portion which is from the human IgG and only the antigen binding region from the mouse origin, then the side reaction is so rejection part is lowered. That's how humanized, and they are all made in the laboratory by recombinant technologies, engineered antibodies, right? So anybody else?
I have one more question actually. While actually Dr. Salunke was talking about we can use that viral DNA and RNA also as vaccines. So how do we ensure the stability of the RNA as a vaccine once it is present in our system?
I think I will let Sanjeev Kumar, who is making DNA RNA vaccines, to answer that question.
Just can you repeat the question? You were mixing up stability of the RNA in the body and in the vial. What is that?
So my question is that Dr. Salunke mentioned that we can use viral DNA and RNA as vaccines. So how do we ensure its stability once it's in the system?
Okay, so what happens is that you have made some modifications to the RNA so that it can stay longer. That's all. Otherwise, it has to be taken up inside the nucleus, or at least RNA has to be taken up inside the cytoplasm almost immediately. Otherwise, it won't work. In the case of DNA vaccines, one of the problems is that there are so many nucleases around that as soon as it enters the body, it gets chewed up. So there also, only when you give it intramuscularly, some of it gets taken up by the nucleus, and that's what that gets expressed. DNA vaccine technology actually did not take up much. I mean, I did DNA vaccines 20 years ago, DNA gene therapy 20 years ago, and had to step out of the field because the field was not going anywhere. Primary reason being that it is unstable in the body, gets chewed up immediately. RNA is the next generation technology. By making modifications, it can stay a little bit longer, enters the cell, and then does the job.
I have one more question, like in conjunction to that. It's like when we were studying about immunology, we were being told that the antigen has to be complex enough to elicit an immune response, right? For example, if I have polyglycine, then it will not be an antigen. So there were similar examples that you cannot exactly also use DNA or RNA for vaccines. So it was a little contradictory to what I have read. So that was also a question.
Discuss outside now, right? Because it's
So you will basically any antigen which is so antigen needs to be a little complex to be picked up, right? So if it is in the hapten form is like it is a portion of the antigen which can react with the antibody but does not generate immune response. Then it has to be complexed with a protein or any other carrier to generate immune response. But as such, most of the peptides, proteins can generate, induce, immune response. So as we will go through the course, you will understand that somebody will discuss all this part and you will get your answer. So keep listening to that. So there is a little bit detailed answer to this. So at the moment, we will not be like to take it, but definitely it's a good question, and we will take care of it. We will discuss it. Okay?
Okay, thank you, sir.
Right. Thank you very much. I think it's time to close because we are already gone beyond time. And I would like to thank Dr. Salunke for sparing his time and giving such a discourse. because this is the beginning of the course, and he has set the right spirit, because a lot of discussion is coming up from the students, so it's very encouraging. Thank you very much, and I hand over back to the organizers for carrying on.
I thank Sir for his wonderful insights. Now I would request Dr. Sunil Arora Sir to felicitate Dr. Salunke. Now I request Dr. Shriram Shashadri to felicitate Dr. Sunil Arora. We request you all to proceed for tea, and please report back by 12:45. There are also exciting games planned for you all, so please enjoy your heart out there.